Unit 3, Genetics, Cell Division, and Molecular Biology. DNA and RNA. Genes are segments of DNA in a chromosome that code for specific traits. DNA, or deoxyribonucleic acid, contains necessary instructions for a cell to build proteins from amino acids. RNA, ribonucleic acid, helps with gene expression and protein synthesis. Isolating the material of heredity. After Frederick Miescher plus Phoebus Lever did their roles in discovering the two nucleic acids, Thomas Hunt Morgan used fruit flies to provide the first experimental evidence that genes are located on chromosomes. In 1928, Frederick Griffith designed an experiment to study the bacteria that were responsible for a pneumonia epidemic in London. He discovered that the dead pathogenic bacteria could pass on their disease-causing properties to living non-pathogenic bacteria. He called this the transforming principle. In 1952, Alfred Hershey and Martha Chase used radioactive labeling to show that genes are made of DNA. They used a virus that contains a protein coat surrounding a length of DNA. This virus attaches to a bacteria cell and injects genetic information into the cell. The infected cell creates new viruses and bursts, which releases the virus to infect more cells. Hershey and Chase created two patches of the virus. In one, they labeled the protein coat with radioactive sulfur. In the other, they labeled the DNA with radioactive phosphorus. The radioactive phosphorus was found in the bacteria, so they concluded that DNA must direct the cell to make new viruses. Structure of DNA. After isolating DNA and RNA, Lever determined that both molecules were made of long chains of nucleotides. Nucleotides are composed of 5 carbon sugars, deoxyribose in DNA, or ribose in RNA, phosphate, and 4 nitrogen bases. The 4 nitrogen bases are in 2 groups: purines, which are adenine and guanine, and pyrimidines, which are thymine and cytosine. In the late 1940s, Irvin Chargaff found that nucleotides aren't in equal amounts in DNA and RNA. He found that the amount of adenine in a DNA sample is equal to that of thymine. The amount of cytosine is approximately equal to guanine. This is called Chargaff's rule. In the early 1950s, Rosalind Franklin used X-ray photography to analyze the structure of DNA. DNA has a helical structure with 2 regularly repeating patterns. Nitrogenous bases are on the inside of the structure, and their sugar-phosphate backbone is on the outside. Many argue that she should have shared the Nobel Prize for discovering the structure of DNA, but she died before it was handed out. In 1953, James Watson and Francis Crick were the first to produce a structural model of DNA. Their model resembles a twisted ladder. Sugar and phosphate make up the backbone, or the handrails of the ladder. Nitrogen bases held together by hydrogen bonds make up the rungs, or the steps of the ladder. The ladder twists to form a double helix. From Franklin's images, Watson and Crick knew the distance between the sugar-phosphate handrails remained constant. The 2 strands that make up DNA are not identical, but complementary, due to the base pairs of AT plus CG. They are also anti-parallel. The phosphate bridges run in opposite directions in the 2 strands. Each end of a double-ended DNA molecule contains the 5' end of one strand and the 3' end of the complementary strand. In a DNA segment, the number of purines is equivalent to the number of pyrimidines because of the base pairing rule. Nitrogen bases always pair in complementary pairs: adenine with thymine, or A with T, and guanine with cytosine, or G with C. RNA versus DNA. The sugar component of RNA is ribose. RNA doesn't have thymine. In its place is uracil. RNA is single-stranded. There are many types of RNA. For example, mRNA, rRNA, and tRNA. DNA replication or synthesis. Replication. DNA has the ability to replicate itself. This is why one cell is able to divide in two cells, and each cell has identical genetic information. Replication takes place during the S phase of interphase. Replication is semi-conservative. Each new molecule of DNA contains one strand of the original complementary DNA plus one new strand. Each new strand conserves half of the original. Replication takes place at several locations along the DNA molecule simultaneously. The steps are described in sequence to better understand the concept. 1. Replication starts at a specific nucleotide sequence called the replication origin. 2. During replication, weak hydrogen bonds that hold bases together are broken with special enzymes called helicases. 3. This creates two Y-shaped areas, or replication forks, at the end of the unwound area. The unwound area is called a replication bubble. The parent strands are conserved, while two new strands are formed, with them as a template. 5. Free-floating nucleotides are attached to the exposed nitrogen bases, according to the base pair rule using DNA polymerase in a process called elongation. DNA polymerase attaches new nucleotides to the free 3' end of a pre-existing chain. Elongation can only take place in a 5' to 3' direction. This means that replication occurs in opposite directions along each parent strand. One strand is replicated continuously. This is called the leading strand. The other replicates in short sections called Okazaki fragments that get spliced together by an enzyme called DNA ligase. This is the lagging strand. Since DNA polymerase cannot synthesize new fragments of DNA, an RNA primer serves as a starting point for elongation. An enzyme called primase is required to construct a primer. When finished the strand, DNA polymerase removes the primer by eliminating the nucleotides in a 5' to 3' direction. Hydrogen bonds form between the nitrogen bases. Special proofreading enzymes, DNA polymerase, check the new strand for mistakes. Errors are removed by cutting the mistake out and using an endonuclease, replacing it with the correct base. As soon as the newly formed strands are complete, they rewind automatically into the helix structure. Replication continues until the new strands are complete, plus the two DNA molecules separate. This is called termination. The replication produces two strands of DNA from one where each strand is composed of half old and half new. Protein synthesis. Genetic code is determined by the arrangement of nitrogen bases within the strands of DNA. Each gene codes for the production of a specific protein. DNA. Transcriptions into RNA, which is translated into proteins. Mutations. A mutation is a permanent change in the DNA sequence caused by mutagens. Mutations are inheritable, arise from mistakes in DNA replication when one nitrogen base is substituted for another, and they create a new genetic code that gives new instructions to make amino acids. Mutagenic agents or mutagens. Physical, pushing/tugging chromosomes. Chemical, carcinogens, mustard gas, poor nutrition. Medications, some antibodies. Radiation, X-ray, UV radiation, cosmic rays. Replication mistake. Natural mistakes occur during mitosis or meiosis. Nutritional. Lacking certain nucleotides in diet means you are unable to provide the proper free nucleotides, plus this causes mismatch. Biological. Most viruses use genetic material of chromosomes to reproduce. They join existing DNA to cause permanent damage. Proteins. Proteins are composed of 20 different amino acids, strung together in nearly endless combinations. Composed cell membranes, cell organelles, muscle filaments, hair and hair color, enzymes, antibodies, and hormones. Genetic code. It takes the code of 3 nucleotides, aka a codon, to code for one amino acid. Humans can naturally make 12 out of 20 amino acids. We need to inject the other 8. Simple protein equals 8 amino acids. A complex protein equals 50,000 amino acids. Sequencing amino acids is determined by DNA. Replacement of a single amino acid can change a protein. The genetic code has 3 important characteristics. 1. It is redundant. More than one codon can make one amino acid. 2. It is continuous. Reads as a series of 3-letter codons without spaces, punctuation, or overlap. And 3. It is nearly universal. Almost all organisms use the same code. Role of DNA in protein synthesis. DNA is in the nucleus, but protein synthesis happens in the cytoplasm on ribosomes. mRNA is responsible for reading information from DNA, transcription, and it carries it to rRNA, where it will be translated into an amino acid sequence by tRNA. RNA, ribonucleic acid. Different from DNA. Single-stranded, ribbon versus twisted ladder. Sugar equals ribose, located throughout. Has uracil instead of thymine. 3 main types: mRNA, rRNA, tRNA. No introns, which makes it shorter. Transcription. Protein synthesis begins with transcription of DNA. DNA never leaves the nucleus. Process. In a DNA molecule unzips in replication. However, RNA nucleotides are now added to necessary areas by RNA polymerase. As the double helix uncoils, nucleotides from the mRNA find the appropriate pair by using single DNA strands as a template, 5' to 3'. Nucleotides bond as normal with the exception of uracil bonding to adenine. mRNA joins and fuses into a long chain. mRNA moves away from DNA, which joins back together. mRNA leaves the nucleus to find ribosomes. mRNA reads code for amino acids, and RNA codes for them. Some codes aren't for amino acids, but rather are initiators, like AUG, or terminators, stop, like UAA, or UAG, or UGA. Translation. Translation is the next stage in protein synthesis. Involves translating codons found on the mRNA into a chain of amino acids to form a protein. Transfer RNA, tRNA, is made up of a single strand of RNA that folds into a clover leaf shape. One lobe contains the anticodon, 3 nucleotides that are complementary to the mRNA codon. At the opposite end is a binding site for the amino acid that corresponds to the codon. Each kind of tRNA has a specific anticodon. Ribosomal RNA, or rRNA, is a linear strand of RNA that remains associated with all the ribosomes. Steps of translation. Translation is activated when an mRNA molecule binds to an active ribosome complex in such a way that the 2 codons are exposed. The first tRNA molecule carrying the amino acid methionine base pairs with the codon on the mRNA, AUG. Another tRNA molecule arrives at the codon next to the first tRNA, and the next tRNA passes its amino acid onto the second one. Enzymes catalyze the formation of a peptide bond between the 2 acids. Ribosome moves along the mRNA strand 1 codon at a time. The tRNA molecule detaches from the mRNA and picks up another amino acid as another tRNA attaches to the mRNA. The process begins with AUG and stops with UAA, UGA, or UAG, derived from the message on the mRNA from the DNA. More about mutations. If a mutation is present in the gametes, it will be passed on to further generations. This is why it is particularly dangerous for pregnant women. Mutation can be grouped under two categories: chromosomal mutations, large mutations that affect numbers or structure of chromosomes, i.e., non-disjunction, Fragile X syndrome; point mutations that alter a single gene. Base substitution. A foreign base replaces the normal one, and each DNA can result in different amino acids being made. For example, sickle cell anemia. Insertion. A base is added into the normal DNA sequence. Frameshift right. Deletion. A base is removed from the normal DNA sequence. Frameshift left. For example, this can cause cystic fibrosis. Translocation. A sequence of nitrogen bases is moved from one location on the code to another. Inversion. Reversal of a sequence of nitrogen bases. Duplication. Duplication of a set sequence of bases twice in one location. Silent mutations. No phenotypical effects because some amino acids can be produced with multiple codons. The body can repair some mutations, but not all. Oncogenes and cancer. In normal cells, protein synthesis is carried out by structural genes only when required. Because protein synthesis is not always required, a regulator gene produces a repressor protein, which switches off protein synthesis and reduces the rate of cell division. Uncontrolled cell division is cancer. Cancer is caused by a mutation due to an environmental factor or carcinogen. Mutation can be a base substitution that prevents the production of the repressor protein, or the movement of a gene from one part of the chromosome to another. If the structural gene is separated from its regulator gene, it cannot be turned off. These genes that cannot be turned off are called oncogenes. The most common oncogene is RAS. RAS is found in 50% of colon cancers and 30% of lung cancers. It makes a protein that acts as an on switch for cell division. Once a sufficient number of cells are produced, it's supposed to turn off. The cancerous gene produces a gene that blocks the off switch, turns the cell into a cancer cell. Diagnosing genetic disorders. Amniocentesis and chronic villus sampling can take cell samples from a developing fetus or embryo that can be screened for certain disorders using a DNA probe that can find problem genes. When a parent finds out that their child may have a disability that they aren't equipped to handle, they may be given the option to terminate the pregnancy. Gene therapy targets genetic causes of diseases rather than their symptoms. DNA vector carries out foreign DNA into the patient's target cells. The DNA vector is usually a virus that has been genetically altered to carry a desired gene. It will eventually transfer the new gene into the cell's genome. There are two kinds. 1. Somatic gene therapy, correcting genetic disorders in somatic cells. Mutations can still be passed on to offspring. 2. Germline therapy, correcting the genetic information in sperm and eggs. Could have many negative effects on future generations, and is banned in Canada. Ames test. Technology to determine quickly, cheaply, and accurately if a chemical is mutagenic. Any mutagen has potential to be a carcinogen. We must test products for their cancer-causing agents, performed on bacteria that have been mutated, so they cannot produce histidine on their own, plated on a petri dish with no histidine, and the chemical being tested, expect no growth. If bacteria are found, conclusion can be made that the microbe has been mutated and is now producing its own histidine. The more colonies found equals higher chance of carcinogenic. Often the chemical is added to liver enzymes because some chemicals are only cancerous after reaching the liver. Some chemicals can cause cancer in some people, but not others. Genetic engineering and recombinant DNA. Genetic engineering, the alteration of an organism's genome by selectively removing, adding, or modifying DNA. Recombinant DNA, the process of cutting out DNA from one genome and replacing it with DNA from another organism, resulting in a transgenic organism. Transgenic organisms, genetically modified bacteria for use in medicine and bioremediation or environmental cleanup. Transgenic plants to improve crop yield and environmental resistance. Cloned livestock and organs for human use. How recombinant DNA works. Use restriction enzymes, endonucleases, to cut strands of DNA within their interior at specific sequences. Then ligase, enzyme that fuses segments of DNA, is used as a biological glue. Production of human insulin. Gene in the human genome that codes for insulin is cut out using endonucleases. The plasmid of an E. coli bacteria is cut so the gene for insulin can be inserted using ligase. Bacteria can read the DNA and produce insulin to be harvested and used. Another example is the insertion of the gene that codes for growth hormone into animals so they can grow faster. Biotechnology refers to the use of organisms or biological products for commercial and/or industrial processes. What are the issues with this? Costs and where money is spent, motivation for the product, biological characteristics of the product, health effects, environmental effects, freedom of information and privacy, and who owns the technology and patents. Cell division. Mitosis, a form of cell division that occurs in all body cells, autosomal and somatic, that consists of one division with 4 phases. The result in the formation of the 2 identical cells. Phases. PMAT. Prophase. Chromosomes form. Nuclear membrane dissolves. Spindle fibers form. Metaphase. Spindle fibers attach to centromere and move chromosomes to a line in the cell's center. Anaphase. Centromeres split, and spindle fibers pull chromosomes apart. Telophase. Cytoplasm splits in half equal to numbers of chromosomes, end up in the daughter cells. Meiosis. A second form of cell division designed to make gametes, sperms and egg cells, that consists of 2 divisions with 8 total phases, results in the creation of 4 genetically varied cells. Meiosis 1. The first division and first 4 phases. Prophase 1. Chromosomes become distinct. Homologous chromosomes, similar size in genes, come together, synapses, during this time, and are so close together that they mix and match genetic code for variation, also known as crossing over. The place where they cross over is called the chiasma. Spindle fibers form. Nuclear membrane dissolves. Metaphase 1. Homologous replicated chromosomes line up on the same spindle fibers. Individual chromatids don't split yet. In humans, since 2n is 46, 23 pairs line up, plus 23 spindle fibers are needed. Spindle fibers equals half of a diploid, equals 1 haploid. Anaphase 1. Homologous replicated hormones separate to opposite poles. Centromeres do not split yet. Telophase 1. Chromosomes arrive at opposite poles. Cytoplasm pinches in, causes cytokinesis in animals, and forms the equatorial plate in plants. Makes 2 haploid cells. Chromosomes may unfold into chromatin and enter interphase 2. Interphase 2 is almost identical to mitosis. Sex linkage. Autosomes. Chromosome pairs 1 through 22, homologous. Sex chromosomes. 23rd chromosome pair. In males, the 23rd pair is not homologous. Females have a homologous 23rd pair. Traits linked to this pair are called sex-linked or X-linked. Genes for sex-linked traits are carried on the X chromosomes, but not on the Y chromosome. Therefore, in a male, the gene on the X chromosome is expressed, whether dominant or recessive. In a female, she must have 2 recessive alleles to have the recessive phenotype. Gender and inheritance. In 1908, Thomas Hunt Morgan did breakthrough work on fruit flies to provide deeper understanding of heredity based on gender. Chromosomes in fruit flies. Females have 4 homologous pairs. Males have 3 homologous pairs. Sex chromosomes are not homologous. Other gender-related inherited traits. Sex-limited. Example, milk production is on an autosomal gene. Bulls carry the genes but don't make milk. The gene doesn't get expressed for bulls. Sex-influenced. Male only needs 1 allele for baldness. Male hormones stimulate the expression of this allele. Females need 2 recessive alleles to be bald. Gene is dominant in males and recessive in females, more common in males. Gene linkage and crossing over. Genes that are found on the same chromosome are said to be linked genes and are inherited together. Genes will not separate during meiosis unless crossing over occurs. Gene mapping. A chromosome map is a linear map of genes on the chromosomes derived from crossover frequencies. It contains the order in which specific genes occur on a chromosome and the distance between the genes. The closer genes are together on a chromosome, the fewer crossovers occur. Genes farther apart have higher crossover frequencies. Crossover frequency is map distance. Pedigrees show traits that are passed on in a family. Circle is female, square is male. Shaded in is affected individuals. Heterozygous individuals are occasionally shaded in. And lined down from a horizontal indicates offspring. Oldest offspring is on the left. Modes of inheritance. Autosomal recessive, for example, albinism. Two recessive alleles is affected, may skip generations. 2 unaffected may have affected offspring. 2 affected may have entirely affected offspring. Autosomal dominance. Anything with a dominant phenotype is affected. Affected only need 1 affected parent. Affected parent can have unaffected children. Sex-linked, for example, baldness. Males are most affected and inherit it from their mother. Females must have affected father.